No sound, Antonio.
So good afternoon, everybody. I hope you hear me well because my computer just died a few minutes ago. Do you hear me well? Because somebody can confirm. Okay, I see Oliver. Okay, good. Thank you. Thank you. Sorry for the inconveniences that I was prepared and now we have these unexpected issues. So welcome this afternoon to this online meeting on new developments for microscopy and bioimaging. It's a pleasure to have all of you here. My name is Antonio Castello. I'm technology manager at EPIC for all the topics related with lasers and also biomedical. And it's very interesting to understand what's happening with microscopy and bioimaging within EPIC. Also to see the work of different different members in components, in solutions. So we are going to learn a little bit of what's happening within EPIC and outside the consortium with these technologies. And today we are very happy to have a sponsor. So the meeting is sponsored by the company Actar Advanced Coatings. So I think Alexander is around here. So Alexander, perhaps you can mute yourself and say a few words about Actar.
Yes, thank you, Antonio. My name is Alexander Teller, heading Actar's German subsidiary. We are specialized in the development and application of ultra black coatings and coated products. And these coatings are used to suppress scattered light and stray light in photonics and optics applications. And we are also active in the field of microscopy and bioimaging, as our inorganic coatings do also have zero autofluorescence. So I'm looking forward to the meeting here, and we are glad to support the efforts of EPIC in this field. Thank you.
Thank you very much, Alexander, and thank you for your effort also sponsoring this meeting. I hope it's going to be interesting also for you. So for the ones of you that doesn't know EPIC, this is what EPIC is: a consortium of more than 800 companies related with photonics technologies, covering very different fields. Of course, healthcare, bio, life science is a very important topic for photonics. Apart from microscopy, very different imaging solutions around both light sources, detectors, new optical components, coatings, as Alessandro just mentioned. So there are a lot of developments that are critical for this topic, and they are within the whole consortium, more than 100 companies and centers related with photonics. And our job as EPIC staff is to help our members. Can be with the technological requests. Can be also with access to new markets, learning about new markets. These technology meetings can help some members to understand what is needed in bioimaging, for example. Also through market reports to understand what's the future and the trends in different markets. We have with human resources activities, with investment or investors that they want to be aware which companies are interested in receive money in different technologies. And of course, networking. That is one of the most important activities. We all have these online meetings to discuss about technologies, to network and understand who is who in this topic, but also physical events. And in this sense, I would like to encourage you to go to our website where we have all the plan for the next months until end of 2025. As I mentioned, a lot of online meetings. Today we are going to talk about technology. The same will happen in late May and June about photonics for computers and AI and photonics for diagnosis and treatment of ophthalmology. But we combine with other topics that are also important for our members. So meeting in human resources, another meeting to discover the career in photonics focusing quantum. Also, as I mentioned, one market report, in this case, micro displays for AR/VR. And technology reports. So we try to offer with these online meetings as much information as possible in different fields to our members. And we combine with the physical events that are one of the strongest activities within EPIC. So we are quite focusing laser wall photonics in Munich, as most of you, I guess, that you will be exhibiting there. So I encourage you to go to our website and register to the different activities we will have there. We'll have a CEO breakfast for all the EPIC members, a marketing coffee to discuss about marketing activities. We will have activities in sustainability as well, also in human resources, and we will combine with different technology meetings. So we will moderate an integrated photonics applications stage. We will have a technology meeting on innovative optics and another one in laser micromachining. And also Carlos will organize a meeting in defense technologies that are one of the most interesting topics in the last months, and a request also from most of you. Before Laser Wall Photonics, we are going to be in the display week this week, in fact, with a panel session. And in June, if you are interested in AR, VR, MR, I encourage you to register to our technology meeting on photonics at MOS Photonics in the Netherlands. But as I mentioned, I encourage you to go to our website and have a look to all the activities that we will have in the next weeks. And related with bioimaging, just to mention two of the meetings that I have organized and I will also moderate. In October, we will go to Switzerland, to Neuchâtel, visit the facilities of the CSEM to talk about photonics for bio and life science, with some focusing pharma, food, and biochemistry as well. And in December, we will have our yearly meeting in medical devices. This year will be focusing cancer pathology, therapy, and surgery. And we will visit Paris, the Gustave Roussy Cancer Center. The goal in this case is to bring medical doctors to the room responsible for different facilities, and understand what's needed from the point of view, what is missing, where are they in the use of the photonics technologies, to discuss with the developers and manufacturers of solutions, and grow together in the way to find a product that will meet the requirements. And we will invite also people that will explain which are the requirements from the hospital management needs. We started last year in ICFO in Barcelona with a very interesting presentation of the process of purchase by a hospital. Once technology is more or less validated from medical point of view, there are several steps that must be taken into account, and I think it's interesting for most of you to understand what's happening in different hospitals, and the same with the certifications and regulations for a medical product. So this is another topic that we will cover in Paris. And for today, we will have interesting presentations, very interesting speakers from companies and research centers. We will cover different work in microscopy and nanoscopy, and also developments in laser sources, sensors. We will also understand what's happening with the multiband pass filters. So I'm looking forward to hear your presentations. I'm sure everybody will be interested in your presentations. So we can move to our first speaker. So Jana, if you can share your screen. Jana Nieder is the research group leader at the INL, the International Iberian Laboratory in Braga, and she's going to present her work with nanoscopy. So Jana, the floor is yours.
Thank you very much for introducing me. Yeah, I'm research group leader at INL. INL is the International Iberian Nanotechnology Laboratory, and today I'm going to talk a little bit about how we can take advantage of nanotechnologies as well as even materials on the atomic scale, such as graphene, to improve or to develop novel type of microscopies. I will not only speak about 2D material or graphene-based applications, but also look into what we can do with other type of 3-dimensional nanomaterials, such as nanodiamonds, and explore quantum effects for advanced imaging. In my research group, we have 3 main research lines, and the research that I'm going to present today are associated to 2 of these research lines. One of them is to indeed develop new advanced bioimaging and sensing methodologies, and the other is to exploit quantum photonics effects or develop integrated photonic devices that have both applications in neuromorphic photonics as well as in quantum information technologies. To give some examples, an overview in advanced bioimaging and sensing, We use nanotechnologies to, for example, develop new type of nanoscopy, near-field super-resolution technologies, nanothermometry, measuring the temperature from within the cell, or to apply nanoparticles in nanomedicine type of topics. I will not have the time today to introduce in these various applications, but I will pick one of them, the near-field super-resolution microscopy or nanoscopy, to speak a little about our recent developments where we are looking into molecular sensitivities. In the area of quantum photonics, you see here mentioned single quantum emitters in 2D materials, as well as nitrogen vacancy centers in diamond. And I'm going to present one of the applications where we functionalize nanodiamonds for a specific neurosensing approach. Last but not least, I will not have the time to talk about the third line of research, the integrated photonics area, where we have quantum integrated photonic devices on the very exploratory level, let's say, developing new technologies for areas such as neuromorphic photonic computing, with an emphasis on spiking nano-LED-enabled integrated photonic devices, a research line led by staff researcher Bruno Romeda, as well as other photonic integrated chips more dedicated to biosensing or to quantum photonics applications. Regarding the work on near-field super-resolution microscopy with molecular sensitivity, I wanted to highlight one of our recent papers where we were able to functionalize a graphene surface with a DNA linker that contains a fluorophore that is completely quenched in its starting state. And upon the addition of the target DNA, we are able to follow the DNA hybridization process by exploiting the near-field quenching in the very vicinity of the graphene that is reduced further away on the nanoscale, so that the fluorescent dye that has been completely quenched in the starting of the experiment is unquenched, and we can detect its fluorescence intensity increasing over time. And the measurements, how they look like, are explained here. We take a recording of wide field fluorescent intensity images, and from this recording, we can see individual spots that are each of them changed over time. So if we follow the intensity spots, and for each of these locations where we have fluorescence at the end of our experiment, we can extract curves where we have first a background level, then the intensity that is due to the background when we perform the pipetting of our target DNA, third, a waiting time until an intensity increase is happening, and fifth, and maximum intensity that is stabilized after a certain time. We can associate these intensity levels to different unfolding statuses of these DNA hybrids, where we have here a fully folded and standing fluorescence DNA hybrid. This was not enough to us to just follow the molecular dynamics, but indeed we contacted our collaborator, Pedro Ipuim, research group leader at INL as well, who is an expert in graphene, with whom we did this work, but we asked him to further develop the graphene into a graphene device where we can perform some electrical gating. Now we would like to move our DNA molecules and not only follow their natural kinetics, but to gate the device and to move in deterministic manner on molecular level the fluorophore up and down. And this is work performed at the time by master student João Acevedo, who is now a PhD candidate, who added DNA origami structures to our graphene substrate and now could follow the motions on demand after a gating of the graphene device. And so the device looked schematically a little bit like this. We have a tetrahedron constructed of DNA, and then we have at different height positions attached a fluorescent label, an atto-488 molecule, and we have the graphene device, which can be gated by a voltage attached to the device. What we can do now is to move the position of our fluorophore from a highly quenched to a less quenched level, which changes the fluorescence lifetime of our dye. And you can read these in more details in this paper mentioned here, in Advanced Materials Interfaces. So what we do now is to look at the interaction that we can have of a fluorophore with the dye in vicinity of graphene, and you can see the distance-dependent quenching and the theoretical expected curve of the fluorescence lifetime measurement here. And we perform experiments first with the test level where we are adding our fluorescent marker at different locations in our DNA tetrahedron origami structure and deterministically. And we work now with DNA origamis labeled with a dye, for example, located at 12 nanometers away from the graphene or at 17 nanometers away from the graphene. And every time we can follow the fluorescence lifetime performance. Before gating the device, we test that our origamis are indeed following the theoretical curve, which you can see here on the bottom right, where we have now our fluorophores attached at these different locations, such as, for example, at a distance of 20 nanometers. And we can see that a 20 nanometer distance is associated to a lifetime measurement of 2 nanoseconds, simply using an inverted microscope, a fluorescence lifetime imaging microscope, where we have a picosecond laser coupled into an inverted microscope, and we detect the photon arrival times for each of these photons, having the ability to measure for each of the pixels in the image the fluorescence lifetime information. As a test, we see that our DNA origamis are indeed intact on top of the graphene using AFM characterization. If we now add our gating to these graphene devices, we can see that we have a typical intensity or also typical lifetime for a fluorescent dye. But if we change the voltage on the gate, we can move the intensity down, and once removing the voltage again, we go to the starting position. So the intensity can be tuned, but we can also see that with increased voltage, the fluorescence lifetime can be changed gradually. We can analyze the effect by the shift of the Fermi level in our graphene device, and therefore have developed a very interesting nanomechanical device that could be used to sense distances. Also, once we have, for example, an interaction with these tetrahedrons and other biomarkers or linkers. So one could imagine that the distance of our labeling dye could be used as a way to measure the height of the entire tetrahedron itself, which may depend on electronegativity or a complex binding mechanism on top of the origami surface. But as I mentioned in the introduction, we would like to move away from only 2D carbon, but look into what we can do if we have 3D carbons. And especially if we work with diamonds, we can think of bright, shining nanodiamonds that contain fluorescent NV centers, and we can use them in future, hopefully, to measure neurosignaling processes. What we need to do is to bring our nanodiamond sensors in very close proximity of axons in neuronal cells. And we do this by adding a functionalization to our nanodiamonds. In this case, via a biotin streptolivine linker, we are moving our nanodiamonds directly to antibody called CAV2.2, which is a channel protein that is quite present in different neurocellular systems. We can now aim to perform optically detected magnetic resonance experiments with such nanodiamonds that are in very close proximity to the neurons, where the action potential, a change of charge, is happening. If this change of charge, which are traveling charges along the neuron, occurs, we expect a magnetic field that is following the neuron. And this is the magnetic field that we would like to pick up through an optical interrogation. But before doing that, we need to make sure that our nanodiamonds indeed carry the intended functionalization that we aim to have. So we test whether we could bind our antibody to our particles. Indeed, our particle size slightly increases upon the functionalization. The zeta potential is still negative enough that they are separated, and they do not change the optical properties, which is great because we want to explore the optical properties for the neuronal sensing later on. We can show that the secondary antibody indeed is labeled with an test dye and overlapping with the nitrogen vacancy emission of the nanodiamonds themselves. We successfully test that these nanodiamonds with their functionalization are not damaging to any of the neuronal cell samples that we have. We worked with the cell line called SHSY5Y, as well as with dopaminergic neurons and hippocampal neurons from mouse models. And we can see that indeed they are biocompatible. They do not disturb the axon growth. So we can see extended axons that are looking very similar between treated cells that were treated with nanodiamonds or with functionalized nanodiamonds. Of course, another control has been made without any diamonds. And also the neuronal excitability is not modified, as can be seen with this calcium ion imaging, where the neuronal signalling is intact. We can show that the targeting, once the nanodiamonds are functionalized with the antibody, is indeed increased. We have an increased factor of 2 to 2.5 to reach the locations of these nanodiamonds in the positions where we can measure action potentials. And we demonstrate that indeed the optical detected magnetic resonance sensitivity of NV centers in the presence of these cellular models can be observed by a simple test experiment where we expose our neuronal cells to a magnet or not. In the presence of the magnet, you can nicely see the splitting of the ODMR traces and could then extract magnetic information from these signals. We have to mention that at this moment, the sensitivity for magnetic field sensing is far from the required sensitivity that we would need to record magnetic fields created by neurons. But for the temperature sensitivity, we reach very relevant ranges that could be interesting, especially in the area of cancer research. We also think that we can increase the sensitivity if we move to pulsed ODMR protocols. What you see here are continuous wave ODMR experiments, which have less sensitivity. With that, I would like to stop and conclude that first thank the students and the researchers involved in the experiments that I showed, and also thank the funding agencies, specifically La Caixa and FCT for a project called Diamond for Brain, as well as national funding via 2 projects, the Quando and the Diamond Connect project, and some cost action project. And you can see here already in the banner that I would like to thank, that I would like to introduce an event where we can discuss advances in bioimaging technologies in much more detail. And I invite all the EPIC members and non-EPIC members, of course, everyone that is interested in microscopy, to join us in November, from the 5th to the 7th of November in Braga, Portugal, where we will be co-organizing together with 2 other institutions, the INL, the ICVS, the Medical School of the University of Braga, as well as the 3Bs, this exciting meeting, where it's just called the Spanish and Portuguese Advanced Optical Microscopy Meeting. Maybe some of you know this event. It is, even though it is Iberian, we have international speakers, invited speakers, and international guests. And we typically have, besides the talks, also technology workshops where microscopy companies have the chance to host technical workshops. So as well as booths at an expo. So I invite you all to participate and to contact me for more information and opportunities if it is having an exposition, sponsoring the event, or participating to learn some of the new trends. Some of the new trends, we will have focus sessions in technologies for 3D models, health and disease model imaging, about AI and microscopy, about new tools for neurosciences, as well as quantum imaging and sensing sessions. And then, of course, the hot topics in microscopy applications and methods, and hot topics in AI and data analysis. And with this, I would like to thank you all, and thank you for the opportunity to share our news here at this epic meeting.
Thank you very much, Jana. So we have now time for questions or for comments. So you can use both the chat that you can find in the menu below. You can raise your hand and make your question directly to Jana. I could perhaps start with a question regarding more the technology, because you know that EPIC is an industrial association. So in this process, in this work, that you have developed different imaging solutions as well in your lab, is something that from a technological point of view, are you missing in terms of light sources, detectors, can be optical components, something that you would like to see from one of our members, for example?
Thinking about now, maybe, loosely related to the work that we showed here, we are interested in single photon emitters in 2D materials or in nanomaterials. So one of the technologies that could be very interesting for us are multi-spot arrays, or detectors that can in every pixel record time correlated single photon counting or photon arrival times that are basically detectors that could be very interesting for us, where we just started looking into what is available in the market. And I think there are still very exploratory technologies available, but they're for sure that could be very interesting for us. That would be one of the things I would like to share.
Very good. I don't know if there is any other comment, any question? Well, if not, thank you very much, Jana, for sharing today. And we remember again the SPAO, the Spanish and Portuguese Microscopy Conference in November, 5 to 7, right, Jana?
Yes, 5th
5
to
to 7
7th
November.
of November.
In Braga. So, well, it's one of our partners events. So if you are interested, you can contact Jana directly, of course, but also feel free to reach me if you are interested in discussing any participation, and we can introduce to the organizers. So yeah, we hope to see you, most of you there as well. Thank you very much, Jana, again for your time.
Thank you.
So we can move now to our next speaker. And it's a pleasure to have here Robert Lytle, group leader at the Fraunhofer IOF, and he's going to present the work with ultra-thin microscopes. So I think it's a quite new concept that they want to present. So yeah, Robert, the floor is yours.
Okay, thank you very much. I hope you can see my
everything good
slides and hear me very well. So my talk will be today about miniaturized multi-aperture microscopes, which we developed here at Fraunhofer during the last years, I would already say. So maybe just, yeah. So our institute is mainly dedicated to all kinds of optical elements and optical systems, and we have a department where we are dedicated to the lithographic realization of micro-optical elements. Here is just exemplarily shown Fresnel lenses, customized Fresnel lenses, beam splitters with anti-reflective coating for ultra broadband spectrum usage, polymer on glass replication, and also lithography on non-standard substrates like these curved mirrors. And then we built from that off micro-optical systems or miniaturized optical systems, for instance, for very popular for illumination, like here, compact array projection, tailored light diffusers to tailor the distribution to certain field of views, miniaturized and ultra-compact imaging systems, very flat. Here amongst them, the flat microscope I will talk about, and then also and multispectral imaging acquisition, where we have different filters here inside a very compact system to get a broad spectral imaging and make such data cubes. So the first question is, can we scale down the optical instrumentation to its final object size? So typically when we have samples in life science, microfluidics, or point of care diagnostic, they have typically a size of, let's say, 1 square inch. But contrary, the instrument for analysis has the size of more a magnitude larger. And the idea is how to compactify this optical beam path here to make it smaller. And of course, you know, the applications where they are needed. For instance, the portable microscopy, also cell monitoring is one target application here. Also in workbenches with where you are working in sterile, anaerobe, or biohazard environments, then you would be lucky to have not such an open system like here, but have a small and compact device, or also digital pathology, where you want to have at least large area scans to be detected. So, and the requirements from these applications are typically resolution. You need at least 1 micron to detect certain features of cells. Yeah, like for instance, Golgi compartments and so on. And you want, or it's needed to have at least 1 bright field, and it's highly appreciated to have 1 or 2 fluorescence channels to detect. And you want to have a large area in the range of a microscopy slide here, this 1 inch times 3 inch. So what can you do? How to reduce the size of an image system? Luckily or helpfully, by increasing the field of view. So the standard microscope objective has a certain magnification, numerical aperture, defining the resolution and also the field of view. One direction is, of course, when you reduce the magnification, then you get a larger field of view, but then you lose also resolution. The other way is the way from lithography. You increase the field of view with a high resolution, then you have to increase these optics. And you can imagine from the lithography systems that you have very large and bulky imaging optics that have a very high resolution to image in high quality a certain field of view. And there's the multi-aperture imaging approach, where you have parallel optical systems which are scalable to the field of view you need, and having here each individual a very small field of view, you have even a simplified optics. So, and this is the way how we shrink our micro optics or microscope imaging systems. The one simplest version is to have a system with unity magnification, which means that in the imaging path, you have a certain simple lens, for instance, and you see a field of view in the object, and for the imaging has more or less the same optical distance. You can image this to a certain sensor, and this have an optical tiling where you have the field of view, for instance, here from certain areas that are tiled and stitched again by optics on the sensor, on the image sensor. And this helps us to scale it to image sensors. Even a full frame sensor of 24 times 36 millimeters could be used here with an homogeneous micro lens array to use it as rather a close up image device, not really a microscope. Because we have just the magnification of 1. Yeah, it's also you can combine this also with top side illumination, that you can even see here the fingerprint, although there is no transmittive light. It's an up or EP mode microscope or close up imaging device. So, but when you want to magnify, then it changes a little. So the optical stitching now is not possible directly anymore, but you need a certain scan, because if you have just this small image and you image it to a larger area, then the next one is not directly adjacent to this anymore. You have not the space here in image in the image plane, and so you have to scan. And there are two different ways to scan, but what you gain here is, with this larger magnification, you can increase also your numerical aperture here, and you have the chance to resolve, yeah, for instance, micro-optical cells and or sorry, biological images like cells and so on. And this was realized with this compact system here. So we have here in this system path everything we need for an imaging and scaling with a certain resolution. We have here an object with a certain cover class topping. We have a working distance of 450 microns. Have a tool with micro lenses here on the bottom is a very shallow one, and here is even an achromatic micro lens on top. And using a fluorescence filter here for also discriminating certain functionalities. Then here is above the image sensor, even with its cover class. So this track length here is approximately 10 millimeters, which is very compact. And this is realized by making these micro lenses with lithography approaches, using also black metal apertures here that we can suppress crosstalk between neighboring channels, and we get a very flat, nearly a plate, which we can then place in front of an image sensor for large image sensors like this full frame with 12 megapixels or smaller one here. And then you have, if due to the increase, it's not a snapshot anymore, but you can use it with a scan. So if you have a magnification of M, magnification factor, then it needs at least M square of scan steps to cover completely the full object. And you have two possibilities. You can make a 1D scan, rather by displacing here the channels slightly with a tilt angle, so that you can cover by scanning linearly each spot of the object. Or you can have it, and this is typically done for smaller systems, using a 2D scan. Then you have to, of course, move in 2 dimensions to cover here. And both options have been implemented here. For the large area of a 12 megapixel full frame sensor, it's rather easier to move the object. Whereas for a small area and a small image sensor, it's also possible to move the optics and the sensor itself, because then it's more lightweight, and this movement could be also made fast. So, and what happens from the data or from the recorded images? If you would have a static image, what you see from multiple channels here, they are arranged in hexagonal arrangement. You have all these channels, and each are taking a certain part of the image of the full frame. And then when moving the sensor or moving the image, then we can lead this to a stitching across the scan dimension. And afterwards, then from software, we blend these images in order to get a flat field correction and a color correction and so on. We consider the filter function for fluorescence filter and so on, and then we get final result, a stitched image which has a shading correction and so on. This is then, of course, not anymore a snapshot, but with scan, it's quite easy to get a full area like these 12 megapixels in a very, very short time. To have now built up the complete system, of course, we need also the illumination here. There are free illumination sources for transmitted light configuration for this microscope, 2 fluorescence wavelength and 1 white for bright view. And we place here, for instance, these microscope slide into, and then we can, with different pattern illuminations, we can then look at the surface and different channels and gain the images from software stitching. Or for the small system here, it's shown it's very, very compact. It has only 3 cubic centimeters of total image optic size, and it's quite easy to have this as a compact and a portable device, for instance, for point of care diagnostics. So with the left, or maybe just go back, with that sample here, or with this system, we received this information here. So we have an array of optical channels, each giving the information from a certain field of view. And by stitching together, I would see it's quite dark. I hope you can see that there is again this scan line here, where you scan multiple in one linear direction, and then from that one could stitch finally and overlap the information from a quite large field of view. So from here, a total field of view much larger than 10 by 10 square millimeters. We have the information of a bright view field of these cells, of these HeLa cells, and also a fluorescence signal of these HeLa cells, and they are overlapped and correlated to each other in a very compact robust device. So my summary and outlook here. So we demonstrated that we can build multi-aperture microscope objective, and these are really miniaturized imaging devices. The typical size here for the demonstrated system has approximately 10x magnification and a numerical aperture of 0.3. So it delivers a resolution better than 1 micron. It's usable with bright field and bright light illumination and also fluorescence modes. The illumination optics is optimized by Fleiss eye condensers and similar to a normal microscope, standard microscope. I showed you that there are 1, 2, or 2D scanning approaches available to cover the complete target size. And we apply, of course, adapted stitching algorithms. And the advantage we have here is, of course, the optics is very compact. We are not much larger than this image sensor itself, maybe 1 centimeter in track width. And another advantage from fabrication is that all the optics is in the end passively aligned, because the high resolution lithography approach for one array already delivers the placement accuracy. So this is really are not only for these microscopes, but also imaging devices we could use here. They can be used in compact spaces and could be applied to difficult workplaces. And one outlook, not shown here too much, is a new device where we increased the numerical aperture of a microscope for 8 channels to 0.65, also with white light source and 3 different fluorescence filters here for an image-based cell sorting line, where the prototype is shown here. And another outlook here is the adaptation for the microscopic concept to a microspectrometer array, where we have here a compact array where we have in each channel 3 local spectrometer data available for a complete multispectral or hyperspectral information. And this is then possible to have more than 30 channels at all available for different analysis at different positions, because this is a fiber coupled concept, and each fiber, of course, could be look or could be pointed to a certain different spot. So the typical epic questions here for the final summarization, what can we do for you, is of course the design and realization for micro-optical beam shapers and miniaturized optical systems at all, and build from that of prototypes of optimized microscopic devices for your specific application. And of course, we also provide design studies, the prototyping, and also the small series fabrication. So what can you do for us is, show us your use case, your specific use case, or your application scenarios, and we can find together a solution for compactification, or and make it miniaturization. We are partners in research projects, and we are looking for compact sensor self-developing kits that we can build prototypes, because often it's like this, that the microelectronics is then larger than the very optimized micro optics. Yeah, so the microelectronics is typically larger than in the end. So if it's possible to have it more compact, then it's would be helpful for us. And that's the end of my talk.
Thank you very much, Robert, and thank you for sharing this information. So now we have time for questions or comments. You mentioned as in the what can you do for us, the partnering in research projects. Can you perhaps describe a little bit kind of projects that you could be interested in having some partners?
This is very, very broad. On the one hand, we can work together in funded research projects, of course, from the German federal to the European federal, or to our federal state funding services. But we also could work with you directly bilateral, or if you have a certain partner, then a multilateral research project or developing project to build and prototype, or to deliver you a certain required necessary optical element. This would be our task in a common, or in a research project.
Mm-hmm. And it was very interesting also to see this array of spectrometers that you presented in your last slide. What's the wavelength range that you want to develop your product?
We have two of them.
Right?
We have two of them. It's from 400 to 800 nanometers. Mm-hmm. So it's rather depending on the sensor at the end. For a silicon sensor, and we also have built but yet not published one for the SVIR using an indium gallium arsenide detector.
Okay, very good.
Yeah, but this was really only an outlook. I would this would be, of course, in separate, could be a separate talk.
No, it was very interesting, because we've seen this connection with the fiber. It looks really, really attractive. So something to discuss in a different conversation probably. Didier, would you have a question?
Yes, I wanted to ask, in the micro-optics that you developed, so you have several diopters that you stack to each other to have a decent image. What are the dominant remaining aberrations of your optical system? Is it chromatic? Is it something else?
Chromatic is especially if you want to cover the complete visible area for if you are more or less narrow banded from a fluorescence or so, then it's not critical. And the rather is not from the optical, but maybe also from the stitching, that you may have stitching artifacts if you it's always a balance between field of view of a single channel and the stitching errors you're doing, maybe from from if you have a distorted field of view for large field of view of a single channel. Typically, this distortion is not a problem, and for this really small field of view, then the optics is that simple that you have nearly or hardly any aberration. So the chromatic is one, I would say, which is dominant, or which is always considered, of course, in the optical design. And this especially if you are expect to have a complete white image, full color white image. Yeah. Then it's
Thank you.
and that's why in the end, that's why we tried to introduce a polymer achromatic lens in the system. Yeah, but it's still prominent because in the end, it's all lenses are are polymer and they have similar dispersion. You are not that free as with glasses.
You cannot use diffractive elements.
Typically, they are not helpful, but they also reduce your resolution and other things, and you have not that space per channel, more or less. The channel diameter is something like 150 to maybe 300 microns, and then placing there a diffractive element, it's you have not that freedom for those diffractive optical elements, because then you have to introduce with each element a certain phase function, and then for certain field of view, then you need also these phase shifts, and then you get those 2 pi jumps, and they get really, really narrow at the outer rim. Maybe only in the one-to-one imaging device, but not with magnification.
Okay, so thank you, Robert. Thank you, Didier, also for the question, and thank you very much for being this afternoon here. And as I mentioned, we can talk in a separate conversation about this spectroscopic array that looks very interesting. Thank you again. And we can move to our next speaker. So we have Oliver Prochenow. He's the CEO of the company Valo Innovations. As you can see in his screen, Valo is part of the Hübner Photonics group. Hübner Photonics is offering lasers, continuous wave, tunable lasers, also femtosecond. And Oliver is going to present how to use this laser for 2 and 3 photon microscopy. So Oliver, the floor is yours.
Yeah, thanks, Antonio. Thanks for the introduction. I think you can see the screen.
Yeah, everything good.
So, as Antonio mentioned, we're working for Hübner Photonics. So Valo Innovations is a part of Hübner Photonics. And yeah, we're concentrating on almost all kinds of lasers in the group. So we can talk about single frequency lasers, narrowband lasers. We're doing tunable lasers, beam combiners, and we are also doing femtosecond lasers in the group. And today I want to talk about lasers, or especially femtosecond lasers for simultaneous 2 and 3 photon microscopy. And I will just show you how it works and what are the benefits. So as a brief introduction, so we're talking about microscopy, we're talking about application in tissues. So the optical microscopy, as you all know, can achieve a very high resolution, a very high contrast, and you can get minimal photo damage and maximum penetration depth. The application examples, or typically ones, are the visualization of tumor structures. I will show you some applications later. You can see cell-to-cell interactions, and you can also do in vivo detection of signaling pathways. And I will just explain you in a few seconds where the benefits of using very short pulses. So traditional or linear fluorescence microscopy is in general limited in the penetration depth and also causes photo bleaching and phototoxicity. And you can overcome these mechanisms by using nonlinear excitation techniques. And I will just show you what we are talking about. So we're talking in general about multiphoton microscopy, which means we want to make use of multiple photons at the same time. So we want to have a look on 2 and 3 photon fluorescence processes. We want to have a look on third harmonic generation, and we also can do second harmonic generation. In general, you can see different things with these different methods. So for 2 and 3 photon fluorescence, you can see cell cytoplasm and elastine. Third harmonic generation is always generated at the change of refractive index. So you are able to see cell morphology, you can see the cell structures. And for the second harmonic generation, you can use it to visualize collagen fibers. As we're talking about short lasers, or as you will see, we're talking about pulse duration in the range of 30 femtoseconds. You can just see that, or you can just imagine that the shorter the pulse, the higher is the probability that you have 2 or 3 more photons at the same time. This is necessary to do multiphoton application because, let's say, only if the photons are exactly at the same time at the same place, these multiphoton absorption can occur. So you see that the probability to having 3 photons is for sure smaller than for 2 photons, and you just need to imagine that the pulse duration is one of the important roles here. You can see here on the left side 2 different pulses with different durations, and I just want to say that the area is more or less the same. So you can see that if you just squeeze the photons in time, let's say, to a very short pulse, you just increase the peak power, and so you get a very, very high peak power pulse. And if you just think about comparing a 200 femtosecond pulse to a 40 femtosecond pulse for 2 photon absorption, the efficiency is roughly 5 times higher if you decrease the pulse duration by 5 times. As for 3 photon processes, the process scales to the square. You can just get a 25 times higher efficiency if you just reduce the pulse duration. If we're talking about having the same 2 photon fluorescence or the 2 photon efficiency, then you are able to also reduce the thermal heating by a factor of 5, because you can just simply decrease the average power by a factor of 5, only by having a shorter pulse duration. And then there are different other, let's say, mechanisms where you can reduce the photo-induced damage, and you get a higher contrast and resolution. Just to illustrate what happens, you can see here on the left side, this is a calibration grid, which was illuminated with a 50 femtosecond pulse and around 5 milliwatts, and you see a very high contrast. If you're just using the same power, or at least the same power, but 160 femtosecond pulse, you see more or less nothing. And this is the main advantage of using really short pulse duration, to increase the contrast in the end. On the right side, you can see, let's say this is a scaled up version of the figure in the middle, but you see that the contrast is even smaller. So if we talk about the 2 and 3 photon or multiphoton processes, the question is, what are the laser requirements? So from our side, I would say you want to have a femtosecond laser with a very low pulse duration. You need to have very clean pulse because you want to avoid the background, which simply heats up the probes. And this also means that you want to have a very high peak power pulse. As I mentioned before, you are able to reduce also average power to get by keeping the same contrast. And this also allows you for long-term imaging of living cells. And this is one of the advantages we have. Finally, you want to have a very good beam profile because you can get a very small focus in the end. And as you know, I think you need to have some kind of dispersion compensation because simply as we're talking about very short pulses, we're talking about a very broadband spectrum just related by Fourier transformation. So the shorter the pulse, the broader the spectrum. So the more dispersion effects play a significant role here. So you need to be able to control the pulse duration at the sample. Yeah, and finally, for integration, you want to have compact systems. There should be no water cooling, so a completely passively cooled system would be fine. And I will just show you that it will work with the laser we have developed. And this is one of the examples what we are able to provide at the moment. We are able to generate around 30 femtosecond pulses with a very, very low background. which, as I mentioned before, corresponds to a very broadband optical spectrum. So we're talking here about a spectrum which covers from, let's say, more or less 990 nanometers up to 1140 nanometers. The output power for this laser is in the range of 3 watts, giving you a peak power of 2.5 megawatts, which is quite significant. As I mentioned, it's an integrated dispersion compensation, so you are able to compensate almost all optics. And yeah, the system is quite compact, has some kind of separate electronics, and it's fully remote controllable and always optimized by itself to the optimum performance. And you can see the very good beam profile. So coming to the question, how we are doing, or how we can do 2 and 3 photon imaging. So as you have seen, the very short pulse corresponds to a very broadband spectrum, and you can see in red that if you take the near-infrared spectrum and do a 3 photon process or a third harmonic, you end up in the range of 300 to, let's say, 390 nanometers. In black, it's shown it's a typical transmission curve of a microscope objective, and you see that there is some kind of transmission, let's say, in the blue part. So you see that after the third harmonic, you are able to detect some light, I would say. If you just compare it to a 150 femtosecond laser, which has a much smaller spectrum, you can see that doing a third harmonic, you end up more or less in the UV, or at least the transmission, the light is not transmitted to the microscope anymore. And this is the main advantage. If you're going with the spectrum above 1080 nanometers, you are able to do third harmonic imaging. And I just want to say, doing this with longer pulses at shorter wavelengths, third harmonic is at least, let's say, it's at least very hard to do. And I just want to show you some examples of what you can do with second and third harmonic imaging. And one example is the high harmonic generation imaging microscopy. And let's say you all know that if you want to do biopsies, at the moment it just takes time. So you want, let's say, in the end, you want to investigate the tissue, and for now there are options that say you take out of the biopsy, you do some kind of fixation, embedding, slicing, and H&E staining, which typically gives you some time frame in the range of 24 hours or more. If you just use the same tissue and put it under the microscope and do second and third harmonic imaging, as I mentioned, you can see all the cell structures, collagen and elastin fibers, and you can get a picture within less than a second and a complete biopsy overview in 7 minutes. So just this explains how second and third harmonic imaging can be used, especially in terms of cancer detection. And it's a very nice and very fast method to do this. As I mentioned before, it's also possible to do long-term imaging with really short pulse durations, simply because you can use very low average powers here. And I just want to show you some examples which were done with the University of Amsterdam, where they did some kind of analysis of brain tissue, where they could follow up neurons for several hours without destroying the samples. And this is one of the advantages of these very short pulse durations. And with this, I want to come to the famous epic questions. What can we do for you? What can you do for us? So at the moment, we would be able to provide demo systems for testing, and we are able to offer solutions for multiphoton applications, not only in bioimaging. I want to mention that the focus is really on making use of very short pulse durations and what you can do with this. And what you can do for us, in general, we are interested in contacts and ideas, and we are looking for collaboration partners who wants to, let's say, explore the technology and to move forward with us. And therefore, thanks for your attention.
Thank you very much, Oliver. So we have again time for questions or comments. Yeah, Didier.
Yes, I have a question. When you go to thick tissue imaging with those nonlinear techniques, you have to collect the photons that are backscattered from the tissue, correct?
Yeah.
So does this bring any artifacts?
For sure, it depends on the optics, and let's say in the end, you are using some kind of different dichromatic mirrors to separate the wavelengths. And let's say, I think in the end, I'm not the imaging expert in the end, but I think that it depends mainly on the optics you are using. For sure, you have to be careful to, let's say, to cover the broad bandwidth.
Thank you.
Justinas?
Hi, thanks for the talk. So the question I had, you had this 3-watt laser, and are you like, is it usually used like 3 watts on the samples, or you end up like
No,
using
you can-
milliwatts?
You can use much less power. So, but in the end, it's, let's say, our experience, it's always a question of power levels. But for sure, you can, or you're using much less power on the sample. And as I said, the very short pulse duration also allows you to do very low power imaging, where you also get a good signal to noise ratio. So in the end, there's plenty of power. Yeah.
And the samples don't get kind of ablated through the very high peak power. So like, as you have a quite low repetition rate, as typical systems I've seen on the market being 80 megahertz or 100.
Yeah. So let's say for sure there is an option. For you need to reduce the average power on the sample, that you do not destroy the sample. But let's say in the end it can give you also to do or it gives you the chance to also to do multi-line imaging and things like this. So yeah, if you just want to use it for let's say a single beam, then it's for sure too much and you need to reduce it, or you can reduce it. Let's call it like this.
And Jana?
Thank you very much for this interesting presentation. I was curious if you used this laser already for the combination of measuring in 3D samples, such as organoids or other, let's say, artificial tissue samples. And if that could be interesting, at INL, we have several groups with whom we are also collaborating that can provide these type of samples, models of cancer or of other brain in the petri dish type of samples. And the other question, as the repetition rate is 30 megahertz, if you already used the laser also for fluorescence lifetime imaging at the same time, so for multiphoton film type of applications.
Yeah, thanks. So let me start with the second question. Yes, we did. It's a simple answer. And for the first time, for the first question, I think that's exactly what we want to achieve with these epic meetings, to work on collaboration. So we would be very happy to get in contact with you and to talk about what is possible and what we can do together.
Okay, fantastic. Thank you so much.
Any other final question for Oliver? Well, if not, thank you again, and thank you for the presentation. And as you mentioned, I hope some collaborations arise from these questions today and the meeting today.
Let's hope. Thanks.
Thank you. So we can move to our next speaker. So we have Michel Antolovic, he's the CEO of the company P Imaging Technology. So Michel, the floor is yours. Imaging is a company that are specialized in single photon sensors, cameras. So we are going to learn today how to use their the SPAD sensors in super resolution microscopy. So Michel, the floor is yours.
Thank you, Antonio, very much for the introduction. It makes the start of my talk easy. Is the sharing okay?
Everything good, thank you.
Perfect. So I'm excited to be today here to share a bit more about how we can revolutionize things with SPAD arrays mostly. So I'm joining in from the beautiful town of Lausanne in Switzerland. We are placed in on the shores of Lake Geneva with also some if you would increase a bit our altitude, we would see Mont Blanc here in the back. What is bioimaging? We are a company focusing on single photon avalanche diodes. So we are a deep tech company with extremely short innovation cycles. That's what we pride ourselves on. we have a very lengthy experience in designing semiconductor chips based on single photon avalanche diodes, and of course then adding optics on top and high-speed electronics to make the best out of the chips. When we talk about typical use cases of SPADs, I think one of them was already mentioned by Jana. It's fluorescence lifetime imaging, but I'm going to also touch upon many other fields. So reiterating the short innovation cycles and the innovation potential, I just want to emphasize the fact that we have recently released 3 products. One is a small SPAD array focused on confocal microscopy with increased sensitivity for the red fluorophores, specifically targeting the Atto and Alexa 647. And then our highlight SPAD Alpha, which is a 1 megapixel SPAD camera. It's a successor of our 512 by 512 camera, the first SPAD camera released in the world in 2021. Finally, SPAD 93 for high dynamic range imaging. To take one step back, I would like to question why somebody would use SPAD sensors. And there's really 2 main distinct features why we would do that. And it's the property to have extremely good image quality in low light conditions. That's just quantified by high signal to noise ratio. So SPADs, they're natively photon to digital transformators, so non-analog noise sources, pure, physics limited statistics. And the other thing is that SPADs are extremely fast. They, because of their high electric fields, they have a timing precision in the range of 100 picoseconds, and because they're digital, we can read them out extremely high speed. What we are focusing at Pi imaging is specifically SPAD arrays. So even though I would talk about SPAD point detectors, point is for us always a small array. This has some very important enabling features for some applications that I'm going to mention later on. If you talk about these 2 distinct features, high signal to noise ratio at low light and then high speed, this maps into particular applications, use cases. So if you talk about low light, then you can go all the way to quantum single particle. If you talk about high speed, which is typically connected to low light, we focus on confocal microscopy, flow cytometry, big field. If you talk about timing precision, it's more related to fluorescence lifetime imaging, and then the end use case application for FLIM is typically, for example, cancer sectioning of brain tumor. As I mentioned, we have 3 product types, so point, line, and image sensor or cameras, with quite a few detectors out there. What I would like to emphasize that we provide 2 different types of information. The first type of information is what we denote as time tagging. For this community, it might be more, you might be more familiar with TCSPC, so time correlated single photon counting. It's the same, except that now, in addition to the time, we also get the position within the pixel array where the photon arrived. So best extraction of data possible. And then the other method that we use to extract timing information is called time gating, where we actually have extremely short exposures, and then we can shift this exposure with picosecond time precision. So this technique is very similar to intensified CCDs, except that we are not doing that with a shutter in front. We are doing that really on the pixel level. Because of that, we're extremely fast. Just to give you a bit of feeling what this can be used for, I'm showing here a video where we have a pulse laser, picosecond pulse laser propagating in water. In water, we dissolved a bit of yellow food coloring to enhance scattering in such a way that we can track the light. But here in the following, sequence, you can really see the pocket of light traveling in water. So really high speed, really nice. And what's important, it's not anymore just bound to scan the images. We can also do wide field imaging right away with the camera. Really nice feature. When we talk about applications, it's low light. So I'm comparing here a scientific CMOS camera at really low light levels, so below 100 photons, even lower than you cannot not distinguish. I encourage you to download our white paper where we compare these two technologies. And just as well as highlight, we are soon going to release as well a comparison between EMCCD and SPAD cameras. Okay, now jumping into the core of the presentation, super resolution microscopy. So we use our SPAD arrays in confocal microscopy, a very special technique termed the image scanning microscopy, where you can reduce your resolution below the diffraction limit, or improve your resolution below the diffraction limit. Here on the left-hand side, you can see the diffraction limited confocal data, and then here, second to the right, you can see the increased resolution image. On top of that, what we did here is, in collaboration with our partners from PicoQuant, to then also extract the timing of each photon, and then despite the fact that we had two dyes which emitted the same wavelength, but they had slightly different molecular timing fingerprints, and we can use this differentiation in time to then separate these two channels. So really nice feature also for spectroscopy applications. If you dwell a bit deeper on how this looks in 3D, the difference is even bigger. Typically, we can talk about 100 nanometers lateral resolution and quite improved axial resolution, keeping the sectioning of the fluorescence sample. If you then extract a bit further and we switch to SPAD cameras, one distinct feature that we are enabling is high-speed fluorescence lifetime imaging. Here on the left-hand side, you can see again a cell which was targeted with two distinct fluorophores with two distinct lifetimes. And I wanted to also show on the right-hand side that this is real time, which is also one of the main challenges that we solve in lifetime imaging, that now we can actually do wide field, so extremely high field of view, and at the same time high speed. Okay. Not to dwell too much on this, I would like to thank you for your attention and would be glad to take any questions that you may have.
So thank you everyone. Thank you very much, Michel, and for sharing all this information. We have again time for questions or comments. Yeah, Robert.
Hi, thank you for the talk. One question about your sensors. What is currently the areal fill factor of the photosensitive area compared to the complete pixel size? So I remember from SPADs that they have a lot of electronics for evaluation and analysis of this signal. And do you use optics to virtually increase fill factor?
That's correct. That's correct. That's also related to what Jana was mentioning before. The data that or the information that you have, this is something that was the case in the past. So currently we are talking about SPAD like normal other detectors. So the sensitive area is as big as the pixel pitch.
Okay. Thanks.
And indeed, it's using specialized micro optics for that.
Okay, thanks.
Alexander.
Yes, thank you. Very interesting talk. Very exciting technology. There are also TCSPC cameras based on MCPs, on multi-channel plates. How does your camera compare to that devices? In terms, let's say, maybe sensitivity, timing, dark counts, things like that.
What comes to my mind is one specific product, which of course I cannot now name, but from what I understand is that the technology based on MCP has one photon at a time capability, which means that, okay, there is a maximum count rate of 80 megacounts per second, and it's, if it's more than that, the sensor gets saturated. For us, all the spots, they operate in parallel, and they're fully independent. So our camera, for example, allows 26 giga counts per second. I would say that that's the main difference, if I understand correctly what's offered there.
So it would be mainly the counting capability, how many photons you can count in total.
Dynamic range, and finally then the frame rate.
Yes, thank you.
We have another question from Didier, please.
Yes, it's regarding the time gating slide that you have shown us. You have frame 1 and frame 2. How fast can we switch from frame 1 and frame 2, and how many cycles, which frequency of the cycles you can get there in each of the frame?
Yeah, this is extremely fast, so it takes us just a few tenths of nanoseconds. So typically, what we do, we have a 20 to 80 megahertz laser, and then we repeat that. We take one image by repeating many of these laser cycles. Let us assume this is maybe 100 microseconds or 1 millisecond. Then we shift the gate by 100 picoseconds. We do the same, and so on. And in this way, we actually reconstruct the decay of the lifetime of the light.
Okay. So you could do a lock-in imaging.
Absolutely. So there is no dead time related to phase shifting.
Thank you.
Any other question or comment? I have a question regarding the application in the intraoperative surgical margins. Is something that you also developed with PicoQuant that you mentioned at the beginning, your collaboration, or in the previous slides, or is something that you can also consider developing with other potential partners in this topic?
So this goes hand in hand with a typical EPIC question. So what we are looking for.
Right.
For us, we are looking for people or for companies who can bridge the gap between technique to end use case. This is the biggest value that we see in partnerships. Of course, I would love to, like previous talkers as well, to connect to collaborators. And then in this combination, we provide the technique and somebody else provides the end use turnkey solution. And that's how we bring the biggest value to the clients.
Very good. So I think there is no more questions for you, Michel. So thank you again for being here today. And thank you for the great presentation.
Thank you.
And we can move now to our next speaker. So it's a pleasure to introduce Oliver Pust, Sales Director of the company Delta Optical Thin Film. I think it's a very well-known company doing different kind of filters, optical filters, also the bandpass filters. And he's going to present their solutions for fluorescence microscopy, also with the power of LEDs in the title. So I'm curious, Oliver, what you're going to present today.
Yeah, thank you, Antonio. Thanks for joining. Yeah, the presentation is about how the development of LEDs over the past years and optical bandpass filters open up for new possibilities in fluorescence microscopy and also other fluorescence-based instrumentation. So something went wrong. I have a black screen now. No, okay, it's good. It's good again.
It's
So
you, but we
the
don't see the
content
picture.
of my presentation is a very short company overview, a little bit about fluorescence technology, and then some words about the basic configurations, how fluorescence instrumentation is used.
We don't see, Oliver, we don't see your slides. I don't know if there is
Oh,
some.
that's strange. Sorry.
No problem. You can try to share again, perhaps.
Yeah.
Stop sharing and share again. You see the camera. We see the camera with your face, but no, not the slides for the moment.
If I press the share button now, nothing happens. Okay, here, let me see. Is it better now?
Now it's loading. Yeah. Now we see.
Yeah. Okay.
Thank you,
Sorry.
Oliver.
It wanted one more confirmation from my end to confirm that I really wanted to share something. So, yeah. Just give me a second. Going back to my view here. Then I will say a few words about the development of optical filters and then how that combination is used in modern instruments designing these technologies and what the benefits and applications could be. Because we do not have so much time, I will share two resources in the chat for you to watch another recording and also to read the article that I show here on the screen. There are a lot more details than we have time for today. Yeah, a few words about Delta. Delta is a company that designs and manufactures optical thin film components for OEM customers. So we focus on high quality and high volume. And we have worked with fluorescence applications since the 70s for fluorescence microscopy for companies like Carl Zeiss and Leica. So everything we do has more or less evolved around fluorescence filters, so we know the requirements quite well. We are a Danish company located in Hørsholm, which is close to the capital of Denmark, Copenhagen. And another area we're active in is, apart from homogeneous filters, which we will talk about today, are continuously variable filters. So, of course, you know that fluorescence technology is one of the cornerstone technologies in bioanalysis. It's because it's one of the most sensitive spectroscopic techniques that can be used. And in point measurements enables the identification of molecules. And if you use it in microscopy, you can also see the movement. And if we do time resolved, also the temporal development of biological phenomena. It's used in wide field technology, microscopy and confocal. We have seen examples of that before. It's used in live cell imaging, flow cytometry, and also in clinical testing. So it's well known that fluorophores emit light at one wavelength, typically longer when exposed to light of a typically shorter wavelength. And the fluorophores are designed to bind to different, to typical target molecules, enhancing their emission. And that emission is directly measured for the intensity of the target molecule. One very prominent example is, of course, widely known also in public, the PCR test for COVID detection, where a fluorophore binds to the DNA, and then that DNA is detected using fluorescence technologies. So we have also heard about quenched fluorophores before. So when the fluorophore is free, it typically does not emit any light, and only when it binds to the target molecule, it emits the strong fluorescence when it's excited by the correct wavelength. There are basically two configurations how fluorescence instrumentation is used. The most simple one is a 90-degree configuration, where we have a broadband light source, an excitation filter to filter out the correct spectrum for the excitation. We have the sample that is illuminated, and we have an emission filter that is meant to pick up only the fluorescence signal on the photodetector. The 90-degree configuration is chosen to emit the direct light from the source that could reach the detector. In fluorescence microscopy, you typically have a 45-degree reflective configuration, where you again have the light source and the filter, and in between the excitation arm and the emission arm, we have a dichroic mirror at 45-degree angle of incidence that reflects the excitation light and transmits the emission light. It's also possible to arrange all these components in one cube that you typically have in fluorescence microscope to make the handling and alignment of the components a lot easier than if they were separated. So why do people think about multi-wavelength approaches? What are the advantages not looking at fluorescence microscopy with only one wavelength? So when you have multiple wavelengths available in your instruments, you can identify different target molecules simultaneously. You can do multiplex detection of molecules, and as such, then analyze more complex biological systems. And of course, then if you can do that simultaneously, it reduces the experiment time. And for immunofluorescence arrays, you can detect multiple proteins or structures in your assay. And it also makes it much easier to probe dynamical processes and functional interactions. And for that, you need a light source that can emit different wavelengths and multi-bandpass excitation and multi-bandpass emission filters. But how can that be done? So in traditional multi-wavelength setups, you can, of course, swap the components manually, which is very time-consuming, and for challenging applications, not feasible. What has been done for decades, basically, is to use manual or motorized filter wheels, where you can switch within tens of milliseconds between different filters, and where you have a limited number of excitation and emission filters on the filter wheel that are then put into the beam path for the correct fluorophore to be investigated. Another option is to use scanning gradient monochromators. They can be adjusted to any given wavelength. It can be very fast within a few seconds, but of course with monochromators you are somewhat limited in the bandwidth that you want to excite the fluorophore with, which might diminish your signal. Another option are continuously variable filters. I mentioned that's another field we're working in for fluorescence microscopy. It's typically used on the excitation side, and that has the advantage that you can independently adjust center wavelength and the bandwidth, which makes it very nicely possible to adjust the signal strength of the fluorophore you're looking at. So LEDs have become one of the preferred light sources for fluorescence imaging or microscopy, because 60 years after their invention, they have now become available at very high brightness and at a large range of wavelengths. So you can reach many different fluorophores with the suited LED. And typically they're also quite narrow band compared to broadband light sources, which also reduces the noise signal. And because you do not need to block so much light in a broad wavelength range, also the requirements on the blocking capabilities of the filter can be a little bit more relaxed. They switch very fast within nanoseconds, so that's of course then ideal for multiplexed measurements. And you can also combine them in a compact setup with excitation filters and beam combiners to make a very compact tunable light source. And if you have a multi-bandpass filter, you can use them to switch on and off for multiplex measurements, several LEDs sequentially. And also compared to traditional light sources, they're very energy efficient and have a very long operational lifetime. So in parallel, over these decades, also the filter technology evolved considerably. In the, let's say, 60s or earlier, filters were very often made of color glass or polymers. And then a few decades later, basic interference filters, that's when also Delta started working in that field, were made with thermal evaporation. These were soft coatings. They were not very durable and required sealing. Nowadays, advanced manufacturing has come a long way. Ion-assisted deposition has been also a workhorse for several decades. Then plasma assisted deposition has become the preferred production technology. And later on, we use, since a few years, magnetron sputtering that makes it possible to really manufacture the most complex multi-bandpass filters that we have seen so far. So with that modern technology, you can customize basically any combination of bands, 4 bands, 5 bands, with very high in-band transmission, very high out-of-band blocking, and very high edge steepness. Here is an example of a recent multi-bandpass filters that we made for fluorescence application. And the curve you see on the screen is a measured curve. So it has, you can see that it now approaches, if you will, almost 100% of transmission in a number of bands. And you can, on the next screen, see that also the blocking is what you expect from a very good fluorescence filter, OD6 or even better. And what you can also see that there's there are very steep edges also in the logarithmic plot where the edges really go down almost in a perpendicular. All these filters nowadays are made on single substrates, so they are very compact, very durable, and they can be customized with modern design technologies and the magnetron sputtering technology to specific fluorophore combinations. To give you an idea how that looks like, not that many years ago, we were impressed when we saw filters with 100 or 200 layers. And with the magnetron sputtering, we have produced what you see on the screen here, more than 300 layers in one side of the substrate. And as customers have become aware of these new possibilities, they were also very quick to require specifications that made it possible, for example, to go up to 500 layers on just one side of a substrate. And of course, if you want to work in microscopy, you not only need the bandpass filter for the excitation and emission channels, you also need the beam splitter that goes along in combination with these two components. And also here you can see what has become possible to do with modern design and production technology. Very high transmission in the passbands of the beam splitter, very high reflection, and again here very steep edges. And have in mind it's at 45-degree angle of incidence, so there's basically or virtually no polarization splitting visible on the edges of the beam splitter. And also these can be customized for specific fluorophore combinations, and again are single substrate solutions which can be combined to very compact optical setups. And this is how it looks like in principle. You have the combination of LEDs. You switch on the LEDs sequentially, and then excite different fluid for every time you switch the LED, and the emission filter, of course, then only transmits the respective emission band of the fluorophore. What is quite nice about that instrument design that has been possible with new LEDs and multi-bandpass filters, it's a no moving parts design. So you have seen all the solutions that are possible before. You always need to move something, either a filter wheel, a grating, or variable filters. And here you can have very robust instruments where no moving parts are necessary. So it's possible to switch between wavelengths very rapidly. As I mentioned, it's no moving parts. It also becomes robust, environmentally stable. All the components, filters, and LEDs have very long operational lifetime, so very durable instruments, also reducing cost of ownership. They have low power consumption, so it's now possible to also make portable battery-operated instruments. That's not that relevant for fluorescence microscopy, but for clinical diagnostic tools, that's quite nice to make it more compact than was possible before. So, conclusion of my presentation. We have seen developments of LED and multi-bandpass filter technologies. These two have converged in the past years and now enable really advanced fluorescence instrumentation solutions that can enhance your application in microscopy or diagnosis. It's very versatile, it's very durable, and enabling multiplex detection of any combination of fluorophores, and so enabling real-time biological analysis. And I think that will open up new possibilities in analysis and diagnostics in biology. So thanks for your attention. I think we have time for some questions. And again, look at the chat. I will share more resources just in a second there.
Very good. So thank you, Oliver. And yeah, we will check the links in the chat. So we have time for questions for Oliver. I have perhaps one question related with the use of the filters. You mentioned both in the excitation and in the emission. But what happened in the case of samples that have like a broadband emission? In this case, I guess you need to select a different filter to avoid interferences or a signal that is not related with the wavelength you want to measure, right?
Yeah, I mean, if you really have fluorophores that, even if you excite them narrowly with the excitation filter, have emission that would span more than one emission channel, you would need to look for an additional filter or find a different solution. I'm not an expert on fluorophores. I'm not sure if that can happen, but you need to be, of course, aware of that. Yeah.
And for this development, do you have any collaboration with a LED manufacturer, or you saw what's happening in the market and then you took the decision to go in this direction? How was the process at the beginning?
Yeah, all we do is motivated by our OEM customers. So we, as I mentioned, we work with well-known microscope manufacturers, and they approached us and wanted to have more powerful interference filters, multi-bandpass filters to offer to their customers. And that actually goes for both the emission filters in the microscope, but also some of the microscope manufacturers have their own light sources. And also for that, we have a big project where LEDs are combined with several bandpass filters and beam combiners.
Yeah. And we can perhaps finish with the equation. Is just present what can Delta Optical do for the others, what can the others do for you, Oliver?
Yeah, what others can do for us is to let us know what you require and what the market demands are. Share your ideas with us for multi-bandpass filters in more modern diagnostic tools, and we can help you then make a more powerful product combining these technologies.
Very good. I don't know if there is any other question or comment. I don't see. So thank you again, Oliver. And I see that you share in the chat already the link. So I encourage everybody to have a look to the information provided by Oliver. Thank you again. And we can go to with our final speaker. So it's a pleasure here to have Heiko Bruning, the CEO of the company CubeDot, that he's very fast and already is sharing the presentation. So CubeDot is a company that is developing micro LED displays with also some applications in microscopy. So we are going to learn a little bit about this application of the product. So Heiko, thank you, and the floor is yours.
Thank you very much, Antonio, for the kind introduction. Well, ladies and gentlemen, thank you very much for staying on the call for such a long time. I know it's already pretty close to the end of the day, so I'll try to make it as short as possible. Now, let me quickly introduce CubeDot for a second if you have not heard about us. So we are a pretty young company, founded 2019. And basically what we do is we have a unique technology to process the gallium nitride material system. And that allows us to produce basically lighting systems and micro LEDs for our beam path And with those beam paths, that helps us to set up microscopy solutions where we are ISO certified. And so far, we have produced more than 1,000 systems that we are spread in the market already, which makes us rather happy. Now, if we look about the advantages of our microscopy solutions that we offer to our customer, that's basically that we always customize the beam path, and with that we customize the beam path to the specific application of the customer. The whole setup requires less space. We always produce products that are mechanically highly integrated and robust. We always try to integrate zoom lens or fluidic zoom lens if required. And then we put in the light sources that we fabricate on our own and filters, for example, for fluorescence. And as you can see on the right-hand side, the product that I will present in the next couple of minutes, there is no button, there is no screw, there's nothing you can touch, so there's nothing that could accidentally be wrongly adjusted. Now, what kind of imaging do we do? It's digital inline holography. I'll share a couple of insights on that technology as well later on. It's bright field, it's fluorescence, and of course it's phase contrast, which I'll focus in the first couple of slides. Now, if we compare our systems to those of other companies, we'll try to make our systems as compact as possible, as small as possible, which helps our customers to also move the system from not only one office to another, but also to take it out in the field. Our systems are always driven by USB power, so you won't need any real power supply. USB power via laptop will always be sufficient for any kind of stuff that we do. What I would like to present today is a very compact setup for phase contrast that we will be launching this year in the second quarter of this year. So you'll be the first one seeing that one. And what we put here on the slide is a very small and compact setup that has an optical beam path here. You can see my mouse here. Has a beam path here on top with camera and with a zoom lens and with the objective lens. Down here we got a motorized stage, and then the stage here is also heated. So if you want to keep your cells at the right temperature, you can adjust the heating temperature. Normally it's 37 degrees. And combining all the technology in one simple setup allows our customers, no matter where they are, and we have the customers that will buy this product. They are, for example, some are based in Brazil, some are based in Germany, some are based anywhere else in the world. And what allows this very compact setup that has no buttons to touch basically allows our customers to get the same results in any kind of laboratory, no matter if it's in the field or if it's in the lab itself. They'll always have the same result because the system is so compact and the system does not have any buttons. So there's basically, for our customers, another main advantage is there's no chance of getting an image that is not crisp, which is quite nice, because our customers usually are not specialists in microscopy or specialists in biology. They just want to work with cells. They just want to have the result. So they don't really want to use a microscope as people use a microscope. Now let's have a quick look at the image quality that it delivers. So even though it's very small, it'll give a frame rate of 60 frames per second, which is, for example, sufficient enough to have a look at those sperm cells here of boa, which are boa sperms. Movement is quite nice detectable, so is the resolution. And what does our setup make it so special? Well, there's no annulus ring required, so it requires less adjustment, or at least no adjustment that is required by the customer. So the system works right out of the box. And compared to a conventional approach, of course, it requires less space. And then it's mechanically more robust because obviously there's not so much that you can adjust. Then a quick look at another product that we put on the market a couple of months ago. It's a very compact setup, again, to observe liquids and microchannels. You can see that thing here is a whole microscope, but again, it's totally digital with the sensor down here, and then illumination will be on top here, and it's made by digital inline holography. So there's only one micro LED in there that we produce ourselves, and we got the one detector, 0 lenses, 100% simple mechanical setup, because there's nothing to adjust. You just put in the micro channel, and then you can have a look at your cells easily. And as you can see, as that one is very robust, you can also take it in the incubator, for example. Also, what you can do with it, it's possible to put it in the sterilizer. So if you put it to 125 or 137 degrees, that's no problem for that setup. Now let's again have a look at the resolution. As we heard before in the meeting, 1 micrometer is basically the resolution that is required for cell imaging. And again, what we have here, the resolution goes down to values of approximately 0.7 microns. And what is brilliant about the system is a large field of view. As you can see, it's 4 times 5 millimeters. And you have, no matter where you are on that field of view, on the 4 times 5 millimeters, you always have the 0.7 micrometers in resolution, which is quite nice. Again, it's very robust, it's compact, it's very hard to destroy. Let's have a look at the cells, or at videos that we can take with it. So that would have been a long-term cell observation on neuroblastoma. So we kind of put that in the incubator, and then just had a look at the cells and how they develop. And obviously, the imaging is only black-white or kind of grayscale due to phase contrast. But again, it's much more easy to have systems like that and put them in incubators compared to conventional systems. Right, and then also what we do is it's not only setting up the mechanical setup of the product, but also the algorithms that go aside. For example, object detection, of course, autofocus algorithm, tracking, cell counting. And what we've done here is we just want to showcase that we've done here some image analysis on sperm cells and classification based on how they move, how fast they move, and where they move. So that also goes to what we produce at CubeDot. Now, what can you do for us? Of course, supply filters, supply optical elements that we might need, or also demonstrate interesting imaging technologies that we might integrate to products. So if you're a university or a Fraunhofer Institute and you have a piece of very beautiful technology, we might want to bring it to the market and integrate that to our products. Now, what can we do for you? We design and manufacture customized microscopy solutions. Also, we do the series production of devices that could go along with our logo. It could also go along with your logo as white label manufacturing. And of course, if you require the software to work with a microscope, we can also produce that. Well, that's it. Thanks a lot for your time. I very much appreciate any question. Thank you very much.
Thank you very much, Heiko. So again, we have time for questions or comments. Yeah, Jana.
Thank you very much for the presentation. I'm curious to know now regarding the last proposal, what can we do for you regarding the design and manufacture of customized microscopy solutions? Would you also be available to develop or co-develop microscopy solutions, for example, with the research organization, and thinking about microscope solutions, for example, in more the quantum imaging area, optical detected magnetic resonance, in the end combining something that is more a conventional microscope, but with, let's say, dedicated analysis of signals, etc. So this would require, on the one hand side, hardware, so device control and hardware upgrades and changes, as well as software developments for data analysis, etc. Could that be something interesting that CubeDot would do?
I believe that could sound interesting, yes.
Okay, thank you very much.
Any other question or comment? Yeah, Robert.
Sorry, just you mentioned that you are doing holography, right?
That's correct, yes.
So this is rather that you don't have any lenses between the object and the sensor. Is it the drawbacks you can overcome with a bright light source, right? More or less, and then but depth of focus is because it's not a critical issue.
That's correct. The depth of focus is not a critical issue because we basically zoom or we stack the focus. We stack the focus in Z direction, so that is not a problem. And basically, what it needs is a rather small light source that we can fabricate on our own, as it's a micro LED.
Okay.
And that makes it work.
Okay. Yeah, thanks.
Didier, yes, please.
There's a question about the micro LEDs. Are you able to make a display out of it? How many LEDs can you put together in a plane?
That's a good question. That depends on when you start calling multiple LEDs a display, right? So currently, if you want to address them individually, it's probably about 200, 300, 400 LEDs that we could put together as a display. So, for example, if you look into digital inline holography and want to go for pixel super resolution, then that will be sufficient. If it's for simple matrix codes you want to address, also that works well. But if it's like a full HD image, then that's what we are currently working on.
Okay, so you have an idea of when it will be ready?
The full HD we are looking forward to get things done by the end of this year.
Okay, it's good to follow.
Mm-hmm.
And keep me updated.
I will.
I can perhaps put you in contact, Didier, with Heiko, so then you can have more news, first-hand news about this development.
Thank you.
Thank you very much.
So we are reaching the end of the meeting, but if there is any final question or comment, can be for Heiko or for other speaker. We still have a couple of minutes. In the meantime, I want to thank all the speakers that we have today for all these very interesting presentations about these new developments. It's very nice to see how miniaturization is going also in one of the directions that microscopy is taking, also the new components that are being developed. So I'm very happy. So thank you, Jana, Robert, Oliver, Michelle, Oliver, and Heiko for presenting today. I want to, I said there is no questions. I just want to finish just sharing, as I mentioned at the beginning, the couple of meetings that we will have in the second half of the year, where also microscopy and imaging will be involved. So keep an eye on our website. And I don't know if I'm showing the right one. I think so. So keep an eye on these two meetings in Neuchâtel and in Paris. And I hope to see you there in one of them, depending on your interests. And yeah, we are close to 5. So thank you again. Thank you to all the speakers. Thank you to all of you. And also thank you for your answers to the poll that you see in the screen, because it helps to develop more interesting meetings in the future and to improve the ones that we are doing. Thank you again. And I don't see any other comment or question. So good afternoon, good evening, everybody, and see you in our next EPIC event.
Thank you and bye.
Thank you. Bye.
Thank you.
Bye-bye.
Robert, thank you very much. Thank you.
Bye.
Bye-bye.